Okay, so welcome Celeste and Kristen for the second time. The presentation today, we will discuss the CHROMagar Orientation, is the chromogenic agar we developed for facilitate the urine analysis. And I will talk a little bit at by the end to another chromogenic media, Candida Plus, that you may want to also try, but in a different set of trials. So, who are we, CHROMagar? So, CHROMagar is a French laboratory. We are in Paris. So, Celeste, I already extend my invitation also to Kristen. When you come to Paris, you have to come to visit our laboratory. We will love to welcome you, to have you discussing with our R&D team. They always are very keen to discuss with microbiologists of real microbiology, real nature. So, at CHROMagar, what we do is we develop chromogenic culture media. This is the only thing we do, but we are very good at that. So all our product range, what you can see in our website or in our brochures, is available in powder form. And in the States, when it's in form of ready-to-use plate, is labeled colorants. Okay, so the Colorex plates are made with our CHROMagar dehydrated media. So all this started in the late 70s by this person, Dr. Alan Rambach. So he was doing his PhD at the Pasteur Institute in the 70s, and he was working on genetic engineering using E. coli for as a platform for his genetic modifications. Because he was not a microbiologist, he was struggling into subculturing and picking colonies. And also, as you can see, he has eyeglasses. So he thought that if he could color the colonies that of the bacteria he was working with, that would facilitate his life. So he tested different chemicals until he found one where he could color his E. coli in black. This is a picture, a Polaroid from that time. It's really his E. coli with the first chromogen. And then he thought that this could be interesting not only for research laboratories, but also for any routine laboratory in microbiology dealing with cultures. So he filed the patent, and that was in 1979. He continued to work in his genetic topics and for another 10 years. And it's only in the late 80s he decided to establish his own laboratory and start developing chromogenic culture media. And in the 70s, nobody was interested. When he proposed his idea, nobody was interested. As you know, today, many companies have already developed a few chromogenic media in the range. But at that time, nobody was interested. So since the patent, we have been trying to develop 1 to 2 formulations per year. Today we have about 50 different chromogenic formulations. Half of them are for the clinical microbiology. The other half is more for food, beverage quality control, environmental testing. We have a full team of R&D. About 1/3 of our team is dedicated to the R&D. So again, any idea of any bacteria, any yeasts that you think we should develop a chromogenic medium to help in the detection, in the differentiation, please share this with us. We are looking all the time to new topics, new subjects that are really a need of a clinical bench. Okay, so please don't hesitate to share with us any ideas you think we should go for. Okay, so how this technology works and what's the difference with the traditional media, for example, the McConkey you are using today for your urines. So this traditional media, you use a sugar and the pH indicator to detect the presence of bacteria. So, for example, here we have a McConkey, we have a lactose-positive bacteria. Lactose is the sugar we have added in the McConkey. And the other side, you have a lactose-negative bacteria. This is basically what you can do with this technology. And, of course, you can use only one sugar. You cannot put 2 different sugars in the plate. That will not work. In CHROMagar, we don't use any more sugar and pH indicators. We use the chromogen. It's this molecule I'm showing you here. The chromogen is composed by 2 main elements. The first one is a substrate. And it's a substrate that we will choose to target a specific enzyme in the bacteria that we want to detect. The second element in the chromogen is just a dye. It's a coloration dye. It's used already for many, many years, exactly the same molecule in the fabric industry. So the chromogen has these two characteristics. When the substrate and the dye are being together, it's a colorless molecule. This is when you look at the non-inoculated CHROMagar plate, it's totally translucent. And the chromogen is also soluble. This is why you can add it in the powder, add the water, and prepare your plates. It's totally soluble. So what happens when you inoculate a CHROMagar plate with the bacteria? So we are in the plate, we are in the agar, we have added our chromogen. Then the bacteria we have inoculated will release an enzyme, the enzyme we want to detect by this chromogenic reaction. The enzyme will recognize the substrate that it will need for one of the metabolic paths in the bacteria, and will cleave the substrate separated from the dye. Then what happens is that two free dyes will bind together in an oxidation reaction, and in this reaction they will get a color and precipitate. Okay, so that's the whole secret of the chromogenic medium. How to make it work is more complex, but that's the principle. You start with the substrate that will target a specific enzyme in the bacteria, and you end up with a precipitate that has a color. So, what are the main advantages of this technology? First is because we target very specific enzymes, we can go up to species differentiation, what of course you cannot do just with the sugar. The sensitivity and specificity of the culture is much, much higher. Again, the specificity you can understand easily because we target specific enzymes. And the sensitivity is also because you can differentiate a pathogen among a very complex flora, which increases the sensitivity of your test. It's very easy to use an interpretation. Of course, learning how to read these plates, it takes just a couple of days. Turnaround time, we are talking about usually next day results with this technology. So, how we can improve your urine cultures with our plate? So, this is to show you how it's the prevalence of the different common pathogens in urine. So we are in front of two cases, uncomplicated UTIs. By far, of course, the most common part is the E. coli, the uropathogenic E. coli. Then you have a Klebsiella, Staphylococcus, Enterococcus. The percentage can vary a little bit from the different patients, but this is basically what you can, and probably also what you see in terms of proportions in your laboratory. Okay, so E. coli, Klebsiella, Staphylococcus, Enterococcus, group is Streptococcus, Proteus, Pseudomonas. So what did we do? We developed a chromogenic culture media where you can differentiate these most common pathogens that I have already cited. Okay, as I said, with the technology you are using today, which is a McConkey and blood agar plate. So in the McConkey, you will get gram negatives, and you will be able to separate between fermenters and non-fermenters. And in the blood agar plate, well, you will have to detect the rest, among other things, the gram positives. We use only one plate for the CHROMagar orientation, again, because it's non-selective, and also you will have the gram positives and gram negatives. One of the big interests of this media is also that the specificity for E. coli, this morphology, these big red colonies, is super high, 99.8%. This means that when you get these colonies, you know it's E. coli, you can skip your identification, your multitroph, and go directly to the AST. Another nice feature of our media is that Proteus does not swarm. As you can see, we have inoculated in this plate Proteus. After 24 hours, the Proteus remained in its inoculation area. It didn't swarm in the whole plate. This immunoculum in a blood agar plate would have covered the plate the next day. Okay, in our medium, it does not. Another interesting feature is also the high specificity for Enterococcus, which is also a very important organism in UTIs. This turquoise blue, this small colonies turquoise blue, is super specific for Enterococcus. And of course, all the family of lactose fermenters like Klebsiella and Enterobacter, which in Maconchi will look the same, in our medium are totally different from E. coli. E. coli is red, Klebsiella will be blue. So even when you have mix, you know that you have a mix, very easily to see. Okay, so how did we get such a high specificity for E. coli in our medium? In fact, we used the detection of two enzymes. One is the beta-galactosidase that we detect with the red chromogen, and the beta-glucosidase that we detect with the green-blue chromogen. So E. coli is positive only for the beta-glucosidase. This is why the colonies are red. The family Klebsiella, Enterobacter, Cytobacter, Serratia, they are positive for both enzymes. So we have a combination of the red chromogen and the green chromogen. This is what gives this dark blue, what we call it metallic blue colonies. Enterococcus is only positive for the beta-glucosidase, and this is why it gives this, we call it turquoise blue colonies. Group B Streptococcus agalactiae, it has a weak glucosidase. And this is what we get, why we get this translucent bluish colony, okay? Very different from the Enterococcus. And how do we know that is super specific? The choice we did for the enzymes is when we look at the API database. The API database is the database that is used in terms of prevalence for all the identification, biochemical identification instruments, like of course, this API strips, but the Vitek, the Phoenix, the Microscan, they are all based in this database of prevalence. So what we see is, for example, the ONPG, which is the substrate to detect the beta-galactosidase. All the enterobacteria, Cytobacter, Enterobacter, Escherichia coli, Klebsiella, Serratia, they are all super positive. When we look to the amygdalin, which is the substrate for the beta-glucosidase, we see that all the coliforms are super positives, but not E. coli. Okay, E. coli is very low prevalence for beta-glucosidase, and this is why it remains red, and the other will remain green and in the red, so it makes the dark blue. So let's summarize the different colors you can get for the most common pathogens. So E. coli, we said super specific, these big red colonies. Enterococcus will be turquoise blue. Proteus is a brown halo and doesn't swarm in the plate. Klebsiella, Enterobacter, Serratia will be these dark metallic blue colonies. Group B Streptococcus is the teal blue. Staphylococcus aureus will have its golden aspect. Cytobacter will be metallic blue, usually surrounded by these red halos. Staphylococcus saprophyticus has this pinkish colony, very cute colonies. Candida, in general, yeast will have these translucent small colonies. And Pseudomonas will have the classical pigmentation, the pyocyanin, pyoverdine pigmentation with a flat color. Okay, so this is for the most common pathogens you will probably, or you are finding in any case in your settings. Some pictures of real urines. So this, for example, is a biplate that will be very similar to what you will be testing. So in this plate, you can see, for example, this urine, you see very easily the coli, these red colonies, and you can see a few Enterococcus contaminating the urine, but you easily differentiate the pathogen and the contaminant. In the blood agar will be more work to see what's happening in there. On the left plate is something similar. That was a Klebsiella, an infection in Klebsiella, but it was also a co-infection with a yeast. You see these small translucent colonies? It was a yeast, Candida. Again, in the blood agar plate, you need a lot of experience to analyze what's happening in this plate. Some other pictures, and these are very, very recent. They come from a study and evaluation that was done just a couple of weeks ago in Kentucky, in the University of Kentucky, where they are evaluating the use of our medium in their system. They don't have a WASP lab, they have a KISS trap. So that's one of the issues, that they have to inoculate 10 microliters. They cannot inoculate less, like the usual 1 microliter. So the plates look very, very dense. And in fact, this is also one of the reasons where chromogenic media is very interesting. So in this urine, for example, how many bacteria would you say? How many different species? Difficult to say. This, even for very experienced, is difficult to say because of the density and because everything looks very, very much the same. When you look at the same sample in our CHROMagar orientation, immediately you see the Klebsiella and you see the E. coli. Okay. I'm going back to the McConkey. Will not be easier to differentiate that you have two different bacteria in here. Another example, in here, well, you see a lactose fermenter. And in the blood agar plate, much more difficult. In fact, this plate is covered with proteus. It has covered the whole plate. So very difficult to analyze. If we look at CHROMagar orientation for this sample, immediately you see the proteus, you see the E. coli, and even you can see a little bit of Enterococcus somewhere. Okay? Again, if we go back to the original sample, I think much easier to read in here. And and as you can see, the proteus didn't swarm in the whole plate. You see a little bit of the halo, the tryptophan deaminase halo, but you see, as you can see, it didn't swarm in the whole plate, while in the blood agar was everywhere. One last example, which is very cute also. No gram-negative, nothing growing in the McConkey, okay. And okay, in the blood agar you have a few colonies of something. How many bacteria, which bacteria do you have in here? Difficult to say. When you look at CHROMagar orientation, immediately you see the big dark blue turquoise is the Enterococcus, and you see the pale translucent colonies of group B Streptococcus. Okay, super important. For example, in women, that you have to report also the presence of group B Streptococcus. In here, you see it easily and very, very much different than the Enterococcus. So, to summarize, what are the nice features of CHROMagar orientation? So the first one, of course, is that we can differentiate by the color the most common pathogens that you will find in your urines. Super high specificity to E. coli, 99.8, meaning that you can go directly to AST, no need of spending your time on IDs. It is non-selective, so it will allow the growth of gram-negative, gram-positives, as well as fungi. Clear identification of mixed cultures, as I just showed you. The proteus does not swarm, and the colonies can be directly used for Malditov, other biochemical tests, or antibiotic susceptibility. We will talk a little bit about this feature in a few minutes. And something that might be interesting for you is also that the plates can be stored at room temperature, especially if you have volumes and not that much space in your cold room. These plates can be stored days and days at the room temperature. Okay, so let me now present you a few papers, studies that have been published, evaluations on our media. Today about CHROMagar orientation, there are probably a couple hundred of publications. I will not go to the 200 publications, I will just show you a couple of them. So the first one is a study that was done in Winnipeg, in Canada. So it's a hospital that adopted the CHROMagar orientation, and they wanted to analyze what was the situation before they adopted the CHROMagar orientation in 2006, then in 2009 when they adopted the CHROMagar orientation, and then 2 years later. And they wanted to measure what happened with their workflow, with their manpower, with their costs during these 3 milestones. So first of all, the conclusion of the paper is that CHROMagar orientation medium streamlined the urine culture process and increased the bench throughput by reducing both workload and turnaround time in their laboratories. So it's a very interesting paper. I really suggest you read, or another one, to see how they did this evaluation, how they get all these improvements. Another interesting figure in their paper is the savings they did in additional identifications. So at that time, they didn't have MALDI-TOF, so the savings were measured more in less biochemical tests, like rapid biochemical tests, or ID cards like LITEC, Phoenix, less agglutinations. So they saw really a decrease. The black one is 2006, 2009 is the year of abduction, and 2011 is 2 years after they adopted the CHROMagar orientation. Another interesting point from this graph is, as you can see, already in the year of the adoption, they managed to optimize the workflow. Like 2 years later, they were already at the same level of optimization. So very, very fast, the laboratory managed to optimize its workflow. Another interesting paper study was done by Dr. Baron from Stanford University. They tested in the early 2000s, it's already more than 20 years ago, the use of CHROMagar orientation. They were using full biplates, blood agar and McConkie. And they wanted to test what would happen if they use, for example, a single plate of CHROMagar orientation. So they tested about 1,000 urines, 25%, a quarter of them were with significant growth. And they, of course, they noticed that they saved about 50% of the time of inoculation time. And more important, about 20% of reduction of workup time. They also estimated what would be the break-even cost for the price of the CHROMagar plate with the savings they are doing by using this technology, and they reach about a price per plate of $1.50. In 2000 today, with the inflation, would be something around $2. Around $2 would be the break-even for the use of a little bit more expensive plate, but doing a lot of savings. Another interesting findings of this paper was the antibiotic susceptibility. So they compare the results of picking a colony from our plate and picking a colony from their blood agar McConkie plate. They tested a combination of more than 2,000 isolates and antibiotics, and they showed that they got less than 0.4% major errors. So 0.4% is really the accuracy of the instrument. They show that indeed you can pick the colony and go directly to do your antibiotic susceptibility, and no need of subculture. The conclusion of this paper is interesting. There are some points I would like to highlight. For example, they said that the benefits of CHROMagar orientation include more rapid turnaround time, decreased labor, cost savings on reagents and labor, and ease of use. But they added something that I think is very, very interesting, and every site doing some evaluation has to take into account this point. They say, many microbiologists have years of experience with familiar media and may not welcome any change. We recommend that the laboratory phasing the use of CHROMagar orientation gradually, possibly in conjunction with blood agar plate, until CLS become more comfortable with the new agar. Okay? So this is, of course, a suggestion that we also suggest you follow. One last paper I would like to discuss with you is an international study that was testing comparing the score results using the Malditov, picking a colony from our CHROMagar media or picking the colony from a blood agar or non-selective media. So it was a big, huge study. There were 6 sites. There was one it was led by a Swedish team, and there was a German team, a French team. There was a hospital in London, a hospital in Canada, in Ottawa. There was a Japanese hospital involved. Very, very big study. So each of these laboratories tested about 100 isolates on 10 different of our media, and they pick a colony from the CHROMagar media and the colony from the blood agar, and they compare the scores they got in the identification with the Malditov. And as you can see in here, super easy, really, the chromogenic media and the blood agar plate had a perfect correlation, okay, with also very, very high scores, okay. They were all over two. There were 2 isolates that were under, but it was the same from blood agar and the chromogenic medium, probably isolates that are at the borderline for the Malditov. But for the big majority, as you can see, a perfect correlation in terms of scores between chromogenic media and the blood agar. So you can go directly to the Malditov from our plate. Now, a couple of super fresh slides. This I took from a presentation that was carried out at the last ASM in Los Angeles just a couple of months ago. It was a presentation done by Dr. Davidson from Halifax in Canada. So they have, like you, a WASP lab, and they adopted our CHROMagar Orientation, and they tested the agreement with their system between no growth, no significant growth, and E. coli. And they got really very, very high agreements between their traditional method and the CHROMagar Orientation. Another one very interesting is from Northwell Health. It was by Dr. Ellis. So in their site, they have a Kistra, and they have also adopted a biplate CHROMagar Orientation and blood agar. And again, they do the same. For E. coli, they report E. coli, and for Enterococcus presumptive, and directly to the AST. They save a lot of time and work just because of the specificity of these 2 organisms. Directly to the AST, which is a big savings in terms of time. Okay, so for the evaluation protocol that you will be carrying out in Albuquerque, what we suggest is that we will target this following objective. So, of course, the idea is to compare the CHROMagar Orientation with your McConkey and blood agar plate. The idea would be to evaluate the sensitivity and specificity of our plate comparing to what you are using today, measure the turnaround time for results and from growth to identification. We will try to compare the clinical workflow, the efficiency on this workflow. We will try also again to estimate the cost, comparing the savings we hope you will do with our plate with the extra expenses you do today using blood agar and McConkey. And very important for us also will be to assess the user experience, how the laboratory felt the use of CHROMagar Orientation in terms of learning to use, easy to read, or difficulties, what you can have. That would be a very important objective of the study. So what we suggest is a 2-step study. The first step is just to get acquainted with the CHROMagar Orientation. So the idea would be for you to plate some well-characterized isolates for you to get an idea of the aspect, the color, size. And this is the kind of bacteria we suggest. It's classical, the common pathogens you find, E. coli, Klebsiella, and Enterococcus. In this first step, we will suggest also that you plate just for a game, like 5-10 urines in both plates, and you compare the aspect, the growth, and numeration, so you feel confident. We would suggest that once you have finished this step, you contact us, you send us pictures, and we can make a small session just to discuss your results in case you still have some questions, some doubts. Would be an interesting session. And then the second step will be really evaluating between 200 to 500 urines. I don't know if you have already defined the number you want to test, but we would suggest in any case around this range of samples. And the idea would be, again, each urine sample will be plated in your McConkey and blood agar, incubated, then and will be, of course, inoculated also in our blood agar and the CHROMagar Orientation. Incubation for 18-24 hours. Both plates should go exactly the same path. But with your plate, you will go for your normal protocol, like doing the ID and then antibiotic susceptibility. For CHROMagar Orientation, we recommend you plus a few identifications by Melitov, just so you feel confident that you can do it directly from the plate. Some antibiotic susceptibilities, again, to feel how it works. And then all this data will be fed into a spreadsheet. Okay, we have prepared some templates of spreadsheets where you can report your results. And when this is done, again, we can have another session to discuss results, analyze what you have got. Examples of the files, Excel spreadsheets we can supply, so you will have an easier way to report. Is this kind where you have, for example, one will be for the CHROMagar Orientation, one file will be for CHROMagar Orientation, where you have a specimen, the date, then you have 3 columns for 3 different columns in case you have mixed cultures and you want to report about the 3 of them. And each case, for each cell, when you want, you will get a pop-up menu that will facilitate for you. You can choose a turquoise blue, dark pink. This can be also customized if you want to change, because you want to add other things, you can do it, no problem. We have also, of course, colony counts. We have, by default, less than 10, between 10 and 100, and more than 100. But again, if you want to change this, no problem. You can customize it. And if you do some MALDI confirmations, identification, you can write them. And in here, any comments you feel like sharing with us. And there is a second spreadsheet for the McConkey blood agar plate with the same specimen date. 2 colonies for the McConkey, 2 columns, colony 1 and colony 2, in case you want to report. Basically, the help would be red for lactose positives and colorless for lactose negative. And for the blood agar plate, we have the 3 hemolysis, alpha, beta, or non-hemolytic. Also, of course, colony counts and identification you will get from your MALDI. Super simple. We have an additional spreadsheet that will be for the samples that you will be taking some time. Okay. So that's it for CHROMagar Orientation. Now I would like to again tell you a little bit about our chromogenic Candida medium, because I understood that you do not use chromogenic media. So this is a very interesting plate. I would really suggest you put in parallel to your Saburo. So, as you know, it's very important to really identify the species, especially because we know that today there is not only albicans that can cause infections. You very often find mixed cultures, and it's not easy to read in a Saburo and see that you have different yeasts. And of course, because of the different path of susceptibility, antifungal susceptibility, it's very important that you can get all what is growing in the plate. So in here, I'm showing you the statistically prevalence of the most common yeast, the European statistics and the American statistics. As you can see, they are more or less the same. There are a bit of changes in some prevalences, but the order is the same. So, of course, the most common one is Candida albicans, then glabrata, tropicalis, crozei, and then you have other yeasts that can also come into game. So what did we do to help laboratories, mycologists in identification of this? Is we developed a medium which is called CHROMagar Candida. In fact, the last version is called CHROMagar Candida Plus, where we can indeed differentiate the most common Candida species by the color. So albicans is green, crozei is blue, tropicalis has this pink with a halo, glabrata is mauve, and why we call it plus? Because we also added a very specific morphology to Candida auris, which, as you know, is becoming a huge problem worldwide with infections and also outbreaks in hospitals. Of course, all this would be very difficult to differentiate if you are using only Saboro. So, whenever you feel ready, we'll be more than glad to send you some plates that you can play with this CHROMagar Candida Plus. Just one last slide to show you other media that we have developed at CHROMagar. One for group-based Streptococcus pyogenes. This is for strep swabs. This orange color is directly the PYR test. It is embedded in the agar. This is why it's so super specific. Pink, group-based strep. We developed another one which is also to differentiate C and G. So A will be this orange brown, and C and G will go in pink. For Burkholderia cepacia, that's of course very interesting for respiratory specimens from cystic fibrosis patients. CHROMagar Staph aureus, well, general, everywhere where you have an infection of tissue, you will suspect having Staph aureus, and this is a very, very specific medium. CHROMagar serratian, CHROMagar Helicobacter. As I told you last Friday, this medium was tuned also with the help of Dr. Kulbrith when she was a young PhD at Chapel Hill. They asked us to develop a medium specific for Helicobacter, and we worked that topic for them. And of course, enterics. All the enterics, Salmonella, Campylobacter, Shiga toxin-producing E. coli, Stex, Clostridium difficile, and even for Yersinia tropholytica, where we can differentiate the pathogenic biotypes from the non-pathogenic. The pathogenic will grow in red colonies, the non-pathogenic and tropholytica will grow in blue. Very, very nice video. Okay, one last suggestion is that you visit our website. It's full of technical data. For each product is a full page. There are all the papers that you can, that we found, in any case, in the internet that have been published. You can download them and read more if you're interested in a particular aspect of any media. You will have a lot of data in our website. That's it. That's what I wanted to tell you. And thank you very much for your patience again.